So, I welcome our All India President, Dr. Arvind Deshmukh, sir, our respected Dr. Halami, sir,
Thank
our
you.
today's respected speaker, Dr. Swatantra Kumar, sir, Sally, madam, and all the present participants. Today, we are very fortunate that there is a little bit change in the format of the roundtable discussion. So, today, with us, very renowned scientist, Dr. Swatantra Kumar, sir, is present here. Dr. Swatant Kumar sir is the microbial biogeochemist with international research and teaching experience across the India, Germany and the USA. Currently serving as the associate professor and senior research scientist at Rayat Bahara University, India. And his training includes a doctorate degree in the biogeochemistry from Friedrich University and the Max Planck Institute of the Biogeochemistry, Jena, mainly focusing on the subsurface microbial nitrogen cycling. And he also did MTech in biotechnology, MSc in biochemistry, and all these MTech, MSc, all with the first class honors. He has worked at the Pacific Northeast National Laboratory, the National Center for Polar and the Ocean Research, and the CSI National Institute of the Oceanography, Chandigarh University, with research spanning nitrogen and the sulfur cycles, rhizosphere processes, toric zone priming, and the Arctic and the marine biogeochemistry. Apart from the academic degree, he received also the means Euro 55K competitive research grant from the International Max Planck Research School and has a strong publication record in the journals such as the Frontiers in Microbiology, FEMS Microbiology, Ecology, Oceanology, Metabolites, Frontiers in Water Analytical and the Bioanalytical Chemistry, and serves as a reviewer for high impact journals, including the scientific reports. He also worked in the soil biology and the biochemistry, biogeoscience, and the FEMS microbial ecology. He has coordinated the academic and the examinations at the Chandigarh University, mentored a PhD scholar, and remains actively involved in the international collaborations and the scientific outreach through the conferences, invited talks, and the organizing roles. So we are very fortunate that today with us such eminent personality is present with us to share his experience or his knowledge with us. Before that, I would like to request our All India President Arvind Deshmukh sir to say a few words. Please, sir.
One
Hello.
minute. One minute. Yes. 1 minute. Hello.
Yes, sir. You're audible, sir.
Yes, sir. I'm very delighted today. Such an eminent personality. And moreover, the title of the today's lecture, I'm very much attracted. Right from 17th February, 17th century to 17th February. Very attractive title. And he is covering all recent trends in the life science. I appreciate, I express my thanks to him for coming on our platform. Our lecture is being recorded by the Prathamesh, and that will be circulated again throughout the India. So more students will take advantage in the future. Thank you. Thank you.
Thank you so much, sir, for your kind words. So now I request our eminent speaker today, Dr. Swatantra Kumar, sir. Are you here, sir?
Oh, yeah. Very good
Thank you
evening,
so
sir.
much, sir, for joining our today's session. So now I
Great.
would like to request you to deliver your talk on the topic. So now it's over to you, sir.
All right. Yeah. Very good evening to everyone, and thank you for having me today. It's really my honor, as Dr. Tiwari sir said, and Dr. Arvind Deshmukh ji, yeah, Dr. Umesh Shaili Sharma, who actually introduced me to this platform, and Dr. Sunil, through whom actually I joined this platform. And I'm really honored actually today to present some of my work yeah, with you all. And I just want to say, if there is any issue, if you're not able to hear me, if some technical difficulty or some problem, please interrupt me. Yeah, don't worry, interrupt me, and then we'll try to figure it out. Yeah. So can I share my screen?
Sure, sir.
Okay, great. Thank you so much. Uh, and I also would like to welcome our budding scientists. And there are so many senior people right now in the attending to this talk, and I'm really thankful and honored to be with them. Thank you so much. I will share my screen. Just a minute. Think I don't know, but something happened, and now I'm not able to see anything.
But it is on screen, sir, now. Your screen is shared, sir.
Okay, this is sound. Okay. Great. All right, so that's good. Let me see if I will go change my slide. Are you able to see the slides changing, sir?
Yes, sir. It is visible to all of us.
Okay, super, great. All right. Yeah, so the day uh, Shelley asked me to present the work, and I was so so overwhelmed, and I thought, okay, what could be the topic of the talk? Because I have changed so many different streams. I was able to get you know interaction. I was able to interact with different scientists across the world, and sometimes I'm getting also myself lost. So what would be the best thing to discuss? But yeah, here I am. So from the 17th century to 17th February, that is today, we will discuss new frontiers of microbial life. And as Sir Deshmukh sir had mentioned, I will try to focus, how this field of microbiology, started, and where are we now, actually. So a lot of people have contributed to this microbiology, and yeah, with due note of thanks, I think, yeah, we should proceed, if you allow me to do that. And I welcome to our budding scientists who are students of bachelor's, master's, I don't know, maybe our PhD students also, or some postdoc. So I welcome you all. Yeah, let's begin with the journey. And with that, I will like to acknowledge, some of the places or some of the wonderful people I have met in all these different organizations in India and abroad. Uh, and without their contribution, I was not able to, study more in microbiology. So I'm thankful for all of these organizations and the people who are associated with them. So I will skip this slide because, yeah, as my introduction has already been given, but yeah, here's just a quick note on, what I have gone through in my professional career, so my timeline. So to give you understanding that life is ever changing and we should not be stagnant, and we should always be evolving with the flow. So yeah, great. So, uh, talking about discovery of microbial life, today we are again in this platform expanding the unseen world, from the first sighting of microbes to first mapping and engineering of entire microbial community. And so here I will talk about little bit about the history of microbiology and also some of the work which I have done in during my PhD. Yeah, so we'll look forward to this. So I will start with a little activity, what we know about microbiology. So anybody, we all are microbiologists sitting over here, to be very precise. Let's, draw any picture or just write what we know about the microbiology in one line. So I will come back to you, yeah, with that, what you might have written, or maybe I will pause for a while and ask anybody from the audience how they can define actually microbiology in one line from their own perspective. So I'm taking a pause so that we can interact with our audience. Anyone from the audience, maybe preferably I will say students, master students or bachelor students, they can talk if they can, they can just unmute themselves and they can, say something about microbiology in one line. Anyone?
It is the study of microorganisms and their interactions.
Okay, great. Yeah. Yeah, it's a study of microorganism and their interaction. Perfect. Yeah. Thank you. Anyone else also would like to show some, highlight some important points from boys? Okay. Anyone, any boy or any girl, other girl, they might want to share their views on microbiology in one line.
Study of microorganism
Okay.
and their application in the society.
Okay, great. Wonderful. Yeah,
Two
there was...
definitions are there in the chat box also.
Okay, great. All right. So yeah, microbiology, I think what we know about so many things, so many definition of microbiology from different textbook, from our own experiences and what our teacher have told us, so many different things. With what I'm trying to show you now is a picture of electron microscope. This is the first commercial electron microscope constructed by Hans Ruska in 1939. He was a German physicist who invented the electron microscope. And I would like to highlight his quote, the light microscope opened the first gate to microcosm. The electron microscope opened the second gate to microcosm. What will we find opening the third gate? So Hans Ruska, actually, when he got the Nobel Prize in Physics in 1986, before dying, 2 years before, he made this statement in 1984. Yeah, so what is like, we know that electron microscope, what he made, surpassed the resolution capability of light microscopes. And his invention revolutionized scientific research, allowing for the study of viruses, protein, and also atoms. So when we talk about microcosm, what is microcosm? In microbiology, a microcosm is a small-scale, simplified, controlled model of a natural ecosystem used to study microbial interactions, community structure, and the environmental processes. These self-contained laboratory-based setup mimic natural habitats to analyze complex ecological interactions under specific conditions, such as nutrient additions or contamination degradation. So, they allow scientists to test hypotheses about the effects of, for example, environmental perturbation like pollution or climate change on microbial community in a way that is impossible in the field. And here is the book. So this is the 17th century, which was a rich period for discovery in science and human knowledge. And Robert Hooke, as we all know, he published his book. This is the first book called Micrographia. It was published in 1665 and one of the most iconic books of 389 pages. And it is the prestigious collection at the Royal Society. It gives the reader a glimpse in the field of microscopy, which was rapidly developing at that time. And we know that Robert Hooke, he coined the term cell. The book reproduces the almost frantic series of observation made by Hooke in 1663 and 1664 as a young scientist. He was still in his 20s when he made, when he wrote this book. So Micrographia, this book illustrated with 38 illustrations. To this day, the volume remains a landmark in the history of microscopy. Hooke was a Royal Society curator of experiment at that time and described in an engaging manner how he used his microscope to discover the detailed structure of rocks, plants, and most famously, insects. This was the very first opening of his book. And if you see the left-hand side of this figure, there is a folded page actually very nicely with the first ever microscope actually built and was published, basically, I will say like that. And this is the opened page of that book. And what you can see is description of the early microscope. You can see the light source where the oil is burning. If you look into the figure 5, the K part, there's the oil reservoir and there's a flame burning, and the reflection of that is coming to the lens and on the specimen, and there's a big tube-like structure basically, and there is an eyepiece. And what was most interesting actually, he explained the detailed images of those insects, what he was able to see through those his was microscope. And if you open this, this is so beautiful actually. If we open this whole picture, and this is like 2 or 3 fold picture, and if you open this whole 2 or 3 fold, you can see huge insects. Its image, a very detailed image from that book. And this was written in English. That was also one of the beauty of that time, because most of the time people were writing in Latin. And this was the first time when the English was used, which was a common medium of the communication, so that wide audience was able to understand what these scientists are doing actually in their labs. So this was another figure which was captured. So Micrographia was not only the first book devoted entirely to microscopic observation, but also the first to pair its description with a profuse and detailed illustration. You can see this is very famous and dramatic portrait of a flea and loose, frightening 18 inches long and hardly less startling today than they must have into Hooke's contemporary. So in Micrographia, Hooke clearly describes and illustrates a mold, a fungus, but he does not describe bacteria as we understand them today. He gives detailed structural description, filaments, sporangia of mold, and many microscopic view of plant and animal tissues, but he does not single out free living bacteria or little animalcules. So where bacteria enter the story. So we'll go back a little bit further. Later, Anton van Leeuwenhoek, we already know that, don't know the person. He was the one who carefully described bacteria and protozoa from water, plaque, etc. So Hooke only confirmed Leeuwenhoek's observation of bacteria afterwards. This was the first picture of fungus, drawing of fruiting bodies of a small white spot of hairy mold growing on the cover of a book bound in sheep skin. As you see, the fruit bodies are shown in various stages of descent from HD. Robert Hooke did not spot any spores, but remarked on how fragile the constitution of the pretty bodies was. Then we have Anton van Leeuwenhoek. He built a simple microscope with only one lens, very small, tiny one. It can actually fit in our hand. And he achieved high magnification of about 300x and was able to see bacteria for the first time. So there are three figures in this slide. Figure A showing the replica of Leeuwenhoek microscope. The B revealed its structure, and C showed how it was used. So people had to use really very close to their eyes and see the things, what are there in the sample. So what we can see in this slide is there are two figures. So on the left-hand side, this is from the Micrographia, who gave the first published image of a microorganism or a mold fungus, while bacteria were first described later by Leeuwenhoek and only then confirmed by Hook. So yeah, Leeuwenhoek first observed and drew bacteria, which he termed animalcules, little animals, in a 1683 letter to the Royal Society of London. Using his custom-built, high-powered, simple microscopes, he identified various shapes, wrote spheres and spirals within plaque from his own teeth. His drawing, published in Philosophical Transactions, included rod-shaped bacteria, spherical cocci, and spiral-shaped organisms. And today we know that the cells, we have plant cells, animal cells, fungi, bacteria. Through the microscopes, we were able to see different forms of life or cells. So when we say life on Earth, it is safe to say anything that's made up of cell is alive or vice versa. So let's talk about how much we know about microorganism in today's world, okay? So in the center, there's a big picture of bacteria. So, and then on the left side, there are a few of the, books which we already know of the microbiology. It started with Brock, Biology of Microorganism, then Prescott. There's a, Foster microbiology book, and there's also Totora microbiology book. So these are different books of microbiology which we know today. Which is really. So when I try to grab the what is the information about the microbiology or microorganisms in those books, so I try to highlight what is written over there in a different statement about the microbiology and microorganisms. And I will not go and read each and every line, but I highlighted some key points over here. So let's say microorganism, they all talk about microorganism. They are microscopic organisms, bacteria, fungi, algae, protozoa, and microscopic organisms that are too small to be seen with the naked eyes. Yeah. So, yeah. So then when we see the key aspect of microorganisms, we know that they are diverse. They have human impact. They are microscopic in nature. They are ecological, important, and very important in fundamental processes or so-called biogeochemical cycling. Also, problem which was discovered AMR, so antimicrobial resistance. So generating exchange in microorganisms. So how much we know actually? So do we really know them so well? What we know about them so far? So let's take one few example about the microscopic nature of these organisms. So they say these organisms are too small to be seen with the naked eye, requiring specialized tools like microscope for observation. So in the next slide, I will show you a video. I hope you will be able to see and hear this. If not, please let me know. I will try to share this video in another way. But here it is, the video, which is talking about, microscopic nature of the things which we can, see through the naked eyes and also with the microscope today.
Hello.
डिस्कनेक्ट हो गया है क्या?
I think so, sir. It disconnected now.
17th सेंचुरी पे चले तो नहीं गए नहीं तो?
Where is Dr. Prasanna Kumar? So just I am giving one call to the Sally madam.
Oh, he is back. He is back.
Okay, thank you.
I'm so sorry. I don't know where I missed. So I played the video and I think somehow that time it was connection was broken.
Yeah, you can go ahead now.
Okay. Are you able to see the slide?
Maybe
I'm showing the E. coli bacteria.
It's closed actually, PPT.
Okay, just a minute. I
You
will.
just share your PPT, sir.
Okay. Hmm. Right. Are you able to see my screen now?
Yes, sir. Now it is visible, sir.
I don't know where I lost because I have kept my cell phone also on the side, and I don't know where I was lost in the presentation. What was the last thing which we were discussing?
So it's going to play that video, sir.
Okay, yeah, so that video, okay, I will play that video, maybe the technical problem of the video. So I will share that video in the maybe in the group, and from there people can watch it. So in that video, basically, they talk about different size of the microorganisms, what we know today. They are different size from range in meter to micrometer. So this slide is talking about, the size of the, size variability. It's of the E. coli, which is powerhouse of the bacteria. And we know that it is almost 1 to 2 micrometer long and 0.2425 micrometer in diameter. But bacterial size variability as on 17th Feb, it's not just the 0.2 micrometer or in diameter. There are bacteria which are basically smaller than 0.2, and this is the one of the microorganisms. It is called as Pelagibacter rubic. It was first isolated in 2002 from the Sargasso Sea, from which SAR-11. So Sargasso, so SAR, and 11 is the 11th isolate. 11th number obtained its name. SAR-11 accounts for 25% of all microorganisms with an estimated global abundance of 2.4 into 10 raised to 24 cells. So they constitute nearly, a lot percentage of microorganisms present in our ocean. And on the inside, there's a picture of Professor Dr. Stephen, who is in the Oregon State University, in their group, it was discovered this microorganism SAR-11. And dominates it as though. So the dominance of SAR-11 indicates that it is a critically important player in the ocean carbon cycle, where it is actually working. Its nutrition is actually allotrough, capable of assimilating a wide range of simple growth substrate. So very limited nutrients. Then this is another bacteria. It is called as, Potassium bacteria or ultra small bacteria. So this is a drawing of CPR bacterium. We don't have the cultural representative of this microorganism, so this is a drawing. That's why the names start with candidate. So it is for CPR, candidate phyla radiation. So CPR belongs to a member which are uncultivated, and what we know about them is from meta genomics and single cell sequencing. And they have such a small genome, even they lack certain essential genes to do, their own metabolites, to their own, to sustain their own life. So they're dependent on other microorganisms, basically. And that's the reason we were not able to cultivate these organisms till now. So, and they are considered to be associated with another microbial host cells. Coming next to this slide, it is Thiomargarita magnifica. And if you see the size of this bacteria, these are called as giant bacteria and having length of 1 to 2 centimeter. Yes, you heard me right. They are as big as, a size of your eyelash. So this is one single organism. The bacteria was discovered during the early 2010 by Oliver Gross from the University of French Antilles. He was a PhD student at that time. Initially did not attract much attention because he thought that might be a fungus, and after finishing his PhD, he showed this microorganism to his, postdoc supervisor. And then they started actually working on this microorganism. And the first publication came in 2022, and it was a huge, huge headline, in each and every major journal of scientific publication house. Like, largest bacterium ever found is surprisingly complex. It was published in Nature, a centimeter-long bacterium with DNA contained in a metabolically active membrane-bound organelles. So I mean, it was defying all limits, everything what we know about the microorganisms. It have, DNA packed in, some special capsules. Um, uh, so many different new things which this microorganism told us about. And these are pictures from his paper which was published in 2022. So what you can see here is the size of the microorganisms, and over here you have candidate Thiomargarita magnifica over here. So it's like really like, at the size of your, house fly. You can see over here. And on the inside, you have a, image of a tardigrade, and this is one piece of, this microorganism. And they were able to see what is inside of this microorganism using the TEM, transmission electron microscopy, and they have done higher magnification of the area marked with sulfur granules and papins. The papin actually basically is the point where it is storing its DNA. So it is showing 2 papins. So, and sulfur granule representing that it have a metabolism related to sulfur cycle. So this was a huge thing actually at that time. What I'm trying to show you in the next, there's another video actually. I don't know if this will play nicely or not, but I would like to play this nice video from the Oliver, who is the author of this paper, how he discovered this, big giant microorganism. And I would like to, share this very good analogy of this researcher about his discovery that this microorganism is like, if we are encountering the human being of size of Mount Everest. So this is the analogy which he actually has shared in this video. So I will play this video. Please let me know if I will get kicked out from the Zoom actually again. I hope I will not.
I think that sir has lost his connection again.
I think that video is not possible.
Yes, sir.
You can just explain with the in the form of PPT or these things. Dr. Shelly Sharma can help him, I think.
Yes, sir, he'll rejoin again.
वो वीडियो शायद नहीं चलेगा.
Yes, sir, उनको मना ही करेंगे कि वीडियो ना आप करिए, नेक्स्ट स्लाइड पे आ जाइए.
Dr. Kumar, are you there now?
I don't think so. Yes.
Sir, he has joined now.
Sorry.
Okay, madam.
Sorry, I stopped video.
Sir, Dr. Swatantra, sir, you just don't play the videos. We will
Okay.
share them in the group. You can
Okay.
skip that slide and proceed further.
Okay, great. All right. So I think this is something related to media, multimedia, and that's how it is not, coming through this. So are you able to see the microcosm, which I want to show you now?
Yes, sir, it's visible.
Okay, great. So, yeah, I would like, so after this video, yeah, unfortunately, I was not able to show that properly. I will take you to the next journey of my presentation, where I will talk about, a microcosm assay with microorganisms. I will say enrichment of these microorganisms, which I studied during my PhD time, a long time before. And this is the layout of my microcosm study. So there are some microorganisms which we obtain from some subsurface groundwater wells. We gave them food in the next slide. So I had space gas. We give them food and we sacrifice the microcosm. We collect the sample for molecular work and also for the chemistry. Yeah, so this work basically, yeah, so this work was Sort of, my PhD topic. We published this paper in 2018, which talk about enrichment culture studies as a model of chemolithotrophic denitrification processes in the Heinisch CZE. So this is Heinisch Critical Zone Exploratory. This was in Vienna, Germany. And where in next 15 minutes, again, I would try to give you a glimpse into anaerobic microbial players and processes that are responsible for the removal of reactive nitrogen from a German pristine groundwater aquifers, where chemolithotrophic processes were playing a dominant role. Yeah, so stick with me. If you have any question, please interrupt me if you don't understand, or if you have any any question or suggestions. Yeah. So yeah, I will maybe skip this. I was working with microbial nitrogen cycling, as it's one of the huge problem in the groundwater, excessive amount of nitrogen. As you can see, so if we see different species of nitrogen in the groundwater, nitrate is maximum contaminant level could be 800 micromole per liter. Nitrite should be very, very less, and ammonium also should be very, very less. And if you have more nitrogenous component in the groundwater, there are health implications, especially in the babies who don't develop the denitrifying microbial community, and they will get this blue baby syndrome, also called as methemoglobinemia. So yeah, 3 in 10 people worldwide lack the safe drinking water, and this is a huge thing. Nearly half of the world population relies on groundwater for drinking, and if 3 in 10 people they don't get the proper safe drinking water, it's a huge matter of concern, and especially for our country also. A lot of nitrate is going in the groundwater from the urea, from the fertilizers, and from the industrial waste also. So it is an increasing threat to the limited freshwater resources. And with that, we have side effects such as eutrophication, which is high nutrient in the water bodies. And then we have greenhouse gas emissions and loss of biodiversity as well. If you see the denitrification in the groundwater, 80% of the nitrate is removed in the groundwater by the denitrifying microorganisms, which include archaea, bacteria, and also eukaryotes such as fungi. Anaerobes, they represent nearly 10 to 15% of bacterial population. And this denitrification process, mostly it is done by the anaerobic organism. However, we have seen, there is aerobic denitrification also in the ocean, but there's very limited data from that. But mostly it is 10 to 15% of bacterial population is anaerobic denitrification. So what is happening, it's not like one-to-one reaction, it is the cascade of, different products, which is going through in this very tiny cell. So you have nitrate, which is converted to nitrite, and then you have nitric oxide, and you have nitrous oxide gas, and then the final product is the N2, which is dinitrogen. It is an inert molecule. And this process is mediated by different genes and enzymes, as you can see in the figure over here. So NAR, NIR, NOR, NOS, these are the different genes, and on the top you have different enzymes which are being expressed by these different genes. So if you see, there is difference between incomplete and complete denitrification. So what is incomplete or complete denitrification? So when the nitrate is converted to nitrogen gas, this is called as complete denitrification. But if somehow the conditions are not favorable, it may lead to incomplete denitrification, which means one of the product will form and that's it. Okay, either nitric oxide or nitrous oxide will form, which have huge, greenhouse gas or global warming potential. So there are certain factors which drive this incomplete or complete denaturfication, such as availability of electron donors, such as organic carbon, iron, hydrogen, or reduced sulfur, and the physiological conditions such as pH, temperature, and obviously dissolved oxygen, if most of the denitrification is done by anaerobic organisms. So we studied actually, different wells, and this is a little bit complex. I will try to, give you understanding of my field. So we have aquifers. We have groundwater sample, which is coming from two aquifers. And we have top aquifers and we have lower aquifer. Aquifer is where we have, water. So we have separated these aquifers based on the majority based on the concentration of oxygen and the nitrate and ammonium values. So you can see the oxygen was really, really low in case of the upper aquifer, whereas it is high in the lower one, which is very surprisingly, because if we talk about geology, it is, there are sinkhole through which the water can go very fast. So the upper aquifer was getting recharged with the, rainwater, which was going through the sinkholes, so that bacteria are not able to absorb the oxygen from that water. Whereas the lower aquifer, sorry, I said it opposite. So the lower aquifer was getting water from the sinkhole related system. So the microorganisms were not able to grab the oxygen from the lower aquifer. So the water was more oxygen rich. Whereas the top aquifer, which is the upper aquifer, was very compact, was getting water from the very compact soil. And most of the time, the oxygen was getting, taken over by the microorganisms. So the water was very anoxic. And that was, driving force for different nitrogen component we found in those groundwater. So upper aquifer was having a lot of, upper aquifer was having less nitrogen molecule, whereas the lower aquifer was having more nitrogen-related components. For example, nitrate and ammonia. Yeah. So, and there were very less amount of organic carbon, so which is driving heterotrophic denitrification. So very less organic carbon was available in the groundwater, because most of this groundwater is was basically from the old water through which the carbon was taken up by the microbial processes. So there was very less organic carbon left for microorganisms. So mean that place was not at all party place for the microorganism, for the heterotrophic microorganisms, which mean autotrophic microbial community, they were dominant in that system. So that's what we wanted to see. So if we talk about chemolithotrophy, so the microorganism which can use the carbon from the rocks, from the calcium carbonate, from the CO2, through the Calvin-Wenzel-Beshon cycle, that was making up to 17% of the total bacterial population in that particular groundwater. And that has a potential to fix CO2, okay? And if you look into the different electron donor, so we have thiosulfate, which was having high energy per mole in that, in that groundwater ecosystem, apart from the hydrogen and iron and hydrogen sulfide. So the ability of chemolithotrophic denitrifiers to remove the nitrate in the pristine groundwater was was not clear. It was sort of known, but it was not known how much percentage of these microorganisms in such condition they are able to remove the nitrogen. So research questions like, can we use groundwater originated enrichment culture as a model system to understand the groundwater autotrophic denitrification process and their key players? So to understand that question, we put up 3 objectives. So first is establishment of enrichment culture. So enrichment culture is where you are growing different microorganisms from that particular system in one microcosm. So we got the enrichment culture from the anoxic groundwater. We tested the microbial consortium capacity to use electron donors such as thiosulfate and hydrogen for complete denitrification. And then we try to identify the active denitrifiers in that consortium by sequencing of functional gene transcripts. So the method we use is, for the enrichment, we use different carbon sources. So in that sense, we have CO2 in the headspace and bicarbonate in the liquid media. We have added hydrogen in the headspace. We have added thiosulfate and as an electron electron donor, and nitrate as an electron acceptor, and thiosulfate as electron donor. So we have given actually different food for these enriched microbial community, which have the potential to do the denitrification basically under autotrophic condition. So this is the growth medium targeting cumulative autotrophic denitrifiers. So we have made a recipe of different, different molecules, different nutrients, and apart from this, basically we use a 3 to 6-fold diluted medium than the published literature. People were using very high concentration of these components for their enrichment. But since we were dealing with groundwater bacteria, where there is very less of these nutrients also, so we use very less quantity of these components. All the amount you can see, this is in millimole per liter. All the carbon source, nitrate, thiosulfate, iron, manganese, copper, even the vitamin solution, very, very small, and trace element, very small amount, very small nutrition, like mimicking the oligotrophic conditions. And then we set up the anoxic incubation in dark because groundwater bacteria don't see sunlight. And they were, mostly of the time, they are living in the temperature of 15 degrees centigrade. So we simulate the same environment. And we actually provided different concentration of, hydrogen and CO2. And after 2.5 years of this enrichment culture, we were able to see that with the 5,000 micromolar concentration of those thiosulfate, which is electron donor, and hydrogen, we were able to see clones of enrichment culture. This is 35th subculture, basically, also after 2.5 years. These are the key organisms which were present in our enrichment. So this data is coming from the bacterial 6NS RGNA gene-based MySeq data. So this is the clone library, so which are getting expressed. What you can see is the blue color is, taking more of this column, and that is related to Thiobacillus thiophilus. Next is maroon color, which is Thiobacillus denitrificans. The next is yellow color, which is Sulfuritalia hydrogenovirens. Mean these microorganisms have the capacity to do the mixotrophic denitrification. So not even the autotrophic or heterotrophic, but the mixotrophic denitrification. So then we try different concentration of electron donors in different headspace, and we designed 2 microcosm experiment. In the first microcosm, we gave same amount of, in both microcosm, we gave same amount of nitrate, whereas the electron donor in one microcosm, we gave 2000 micromole, which is very, very less. And in the second one, we gave same amount of concentration, 5000 micromole, which was the concentration of the nitrate, which is the electron acceptor for this process. And then my colleague from IPHT, this was another institution in Vienna, Germany. She measured different gases in the headspace by the Raman gas spectroscopy, such as 15 nitrogen, 15 N2O, and hydrogen gas. And my colleague from Max Planck for biogeochemistry, Einshilk, she measured basically different chemicals from the, in the microcosm, such as nitrate, nitrite, thiosulfate, and sulfate concentration. And what I did over here in this microcosm is I did the most of the molecular work with 16S RNA genes, NARES and NOSG genes, and transcript, qPCR-based analysis, and sequence analysis. So I will be showing very complex, figures, but I will try to explain it. So this is the microcosm 1 experiment. On the x-axis, you can see incubation time in days. On the y-axis, you will see the total gene abundance per liter, and this is for bacterial 16S gene. This is data is coming from the qPCR DNA base. Okay, so you can see the microbial component, they are going up and then get down. So it took a lot of days, like 14 days, to do this microcosm. Okay, and then you can see there's a red triangle which are showing the concentration of nitrate, NO3 negative, during these incubation days. So we started with 5000 micromole of nitrate. So you can see over the another secondary axis where I'm showing the concentration in micromole. So we started with 5000 micromole and on the day 0, and we ended up having nothing, no nitrate in the liquid medium. So most of them actually went either into the nitrite or other component. That's what we'll see next. This is the nitrite NO. Sorry, this will be NO2 negative. I don't know why it is showing NO3 negative, but in orange. Now I'm showing you the concentration of nitrite, which should be NO2 negative. I'm sorry for the NO3 negative. It should be NO2 negative, nitrite, okay? So we saw the buildup of nitrite in our microcosm. The next, we saw the consumption of thiosulfate from the day 0 to day actually 11th. So the electron donor was consumed very fast, and the concentration was just 2000 micromole. So it was consumed very fast, and on 11th day, it was over actually. And we measured the product of this thiosulfate, which was sulfate. So we saw the increasing concentration of sulfate as we go later in the days of the incubation time. So we saw approximate 4000 micromole of sulfate from the 2000 micromole of thiosulfate. And on another side, you can see is the incubation time, which is in 19 days for the microcosm 2. Okay. And again, you have on the y-axis, you have total gene abundance. And in this microcosm 2, we have the ratio of electron acceptor to electron donor is equal to 1, which mean both were having a same concentration of 5000 micromole. Okay, please remember this, okay? In the first microcosm, the concentration of thiosulfate and nitrate was half, approximately. So nitrate was 5000 micromole, whereas the thiosulfate was 2000 micromole. In the second microcosm, we have the same amount of electron donor and electron acceptor concentration. So we saw complete nitrate reduction. Okay, so this is the coming from the headspace. So approximate 2,500 microliter of nitrogen gas from 5,000 microliter of micromole of nitrate, which is perfect. And we saw the complete consumption of oxygen, of hydrogen. Okay, even on the 10th day. So yeah, going back, in both these microcosms, we saw that there is impact of how much electron donor and electron acceptor, how much they are important to do the complete denitrification. Okay, so it doesn't matter that if microorganisms are there, they will do their job. They need proper food, they need proper nutrients, they need a balance of those crucial components, even in the nature itself, to do their job, basically. So then we try to link this information with the nearest gene-based transcriptional activity to nitrate and nitrogen and nitrite turnover. So we see the DNA will form RNA, and from the RNA will make protein. What you can see is the gene abundance. This is the data of qPCR of NAIRS gene, okay, over the time in the microcosm 1, okay. So you will see the high number of NAIRS gene along with the days. So that's the data from the DNA base, okay, and which is correlating very well with our chemistry data, okay. So we have seen high amount of NAIRS gene in the case of 10th days, and also in the 14th day, because we have because in DNA based data there's a limitation of DNA coming also from the dead microorganism. So mean DNA based data sometimes will not tell us the right story, okay, because that is a major problem with the PCR, that it can amplify the DNA from the dead organism also, okay. But here is the next thing, which is very, very crucial. Okay, if you see the red boxes, if you follow the red boxes, we saw the transcript and gene ratio. So you will have the DNA and RNA-based qPCR, and we divide the RNA-based information with the DNA-based gene copy number. Okay, so transcript coming from the RNA and gene is coming from the DNA. So what you can see nicely, so NAIRS was very high in the day 1 because we used the active organism. Then it went down. It tried to adopt the environment. And then again, it shoot up on day 8th. And then it was high up to 10th day. And again, it went down on 11th day. And which is very much correlating with the nitrate consumption, if you see on the right-hand side. Okay, so this figure will show you us the difference between the gene abundance on the y-axis, transcript and gene ratio on the z-axis, and incubation time on the x-axis. So yeah, I don't know if I'm able to convince you that sometime we need to look into the RNA to get the active expression, to see which microorganisms are actually really active, to see the real picture of the microorganism and the ecosystem. So then the next question was, can we identify key active denitrifiers on the transcript level? So which are the key organisms out of our, so many different organisms were there in the microcosm. So can we see those who these organisms are? So we actually took 2 time points on day 3 and day 9, and we made transcript-based clone library. And from that, we can see, based on NAIRS gene and NOS-Z or NOS-Z gene-based transcript library, that most of the organisms are active, which are thiobacillus denitrificans. So the maroon color is showing thiobacillus denitrificans on the day 3. On the day 9, you have less nearest from the thiobacillus, whereas more organism from the nose Z, from the thiobacillus denitrificans, which has been showed as very versatile microorganism which can, do the denitrification in the presence of, a little bit of oxygen also, which can take different electron acceptor, such as your nitrate and nitrite, which can take different electron donors, such as hydrogen, thiosulfate, iron. So this was the prominent organism in our microcosm. So the next question was, can we trace back those key players from the microcosm to the groundwater denitrifying communities? So we took the sample of the groundwater wells. This is me having sampling in the groundwater. And what we saw that this is data which is showing, different microorganisms about the gene abundance. This is the qPCR data. And what we
Sorry
see
to
is-
interrupt, sir. May I
Yes.
request a brief explanation, sir, because most of the students are there, so I have to finish at the 9 p.m., sir.
Yeah. So you want me to quick or you want me explain this?
No, you want to quick, sir.
Okay.
Okay.
Okay. So we were able to see the genetic potential of microorganism in these nitrogen cycling processes. And we were also able to see that 98% of the NARS type denitrifiers in those groundwater, which can oxidize hydrogen and reduce sulfur, which means our microcosm is very much representative of the natural microbial communities. That was the whole thing I wanted to convince to you over here. And the summary, yeah, it's almost last slide. So we were able to solve the mystery of who is driving the microbial nitrogen cycling in the subsurface. Either it is heterotrophic denitrification or autotrophic. And we were able to see that most of the time it's a mixotrophic denitrification, which is actually, working in those oligotrophic groundwater ecosystem, which is converting the nitrate into nitrogen gas. So with this, I will end my slide. So there is need to look at and study biological complexity in much more detail than what is being done currently. Microorganisms that we think are very, very simple might have very, surprising attributes, and we should be ready for those challenging time and those wonderful discoveries. Thank you very much for having me, and the floor is open for the questions. Thank you.
Thank you, sir, for the insightful lecture. So, dear participants, if you have any questions or the observation, you are most welcome. You may unmute yourself now. Or you may write in the chat box also. I may take questions from there.
Okay, I will try to go to chat box.
Very good presentation, actually.
Thank you, sir. Thank you very much.
This denitrification, which you have seen the microbial profiling, right?
In the microorganism, sir.
Yeah. So this was you have done with RNA-seq or only by RT-PCR?
Sir, for that we actually extracted DNA and RNA from the groundwater also in the microbial community analysis, and we did MySeq analysis for the RNA base also. Both DNA and RNA based data was taken.
Yeah. So you did the RNA sequencing also.
Yes, sir.
And this was which year, by the way?
2018.
2018. Okay.
Yes.
Yeah, because RNA-seq was started somewhere in 2007, 2008.
Yes.
So that's why I was curious.
Yeah. So we done.
Just one actually curious as a curiosity. Can you hear?
Yes, sir. Please go ahead.
This Thiomargarita magnifica, you are telling, no, as a
Yes.
gained bacterium.
Giant
How it
bacteria.
has
Sir.
classified
bacteria.
that it is a bacteria?
Yeah, so this is based upon the 6ness RDNA gene. So based on the 6ness gene, which is actually a very prominent biomarker for the bacteria.
Yeah, because you told that.
Initially, it's a very wonderful story that they thought it was a fungus which is
Yeah,
hanging
that is what
down the leaf. And
uh,
when they extracted the DNA, they found out it is not the fungus, but it's the bacteria.
exactly. So, you see that nucleic acid is actually in the nucleosome, means it is
It's in the
encapsulated.
compartment. It is called as papain.
Yes, just like eukaryote, so we can see that.
Exactly. And it also divide by binary division. Yeah.
Yeah, but then yeast also will have that binary division, no?
Yes, but that is not the bacteria.
Yeah,
So the first,
in this case, it is the cystine,
yes. Yeah.
and then they have seen the ribosome also.
Yes, sir.
Wonderful. So you
Yeah.
were in Vienna for the PhD, right?
Yes, sir. I was in University of Vienna with Max Planck for almost 4 years for my PhD.
Which year?
2014 to 2018.
Oh, wonderful.
And after then, I moved to US for my postdoc.
Now, are you continuing the similar work in India?
Unfortunately not, but I'm trying to work with extreme microorganisms. I would like to, share this stage actually for an important discovery which we had made in Chandigarh University. We were able to discover a new species of bacteria which can tolerate UV light for 30 minutes. And some of these isolates basically they can degrade crystal violet, which is one of the carcinogenic, which we generally actually use in all colleges and universities. And we are doing most of the gram staining experiments in the sink. And most of this dye is going to, groundwater or ground surface if there is no proper, I will say, sewage treatment facility or treatment facility present in the university or colleges. So we would try to see if these bacteria are able to degrade crystal violet, and we were surprised to see that in 3 days, these microorganisms were able to degrade 90% of the crystal violet. So yeah, I'm open for any collaboration if some PhD student would like to work on those organisms. These are the novel species. And yeah, we are looking forward for the whole genome sequencing so that we know more about these organisms.
But most of them will be non-culturable, right?
No, these are culturable. These are the cultured bacteria. So they are pure isolates. And that's how, so we did 6ness sequencing and we have submitted it to NCBI also. And they were 70% similar to Bacillus, which mean that novel species.
Very good.
Yeah. So we are open for collaboration. If somebody wants these isolates, we are happy to provide them the glycerol cultures, so cryopreserved cultures. And maybe if somebody wants us from the plate, we can give them the plates. Our next approach actually to do the whole genome sequencing. For that, I was lacking funding. Yeah, and I think MBSI platform is really great. Maybe there are so many big shots sitting in this session right now, and maybe we can put forward a platform, a project actually together where we can work on the whole genome sequencing of those at least 1 or 2 organisms which are very promising.
Any more question from the participant? Salami sir, I think that one question is for you.
For me or
Is
for
any method for identifying the fungi which is poisonous from the organomushroom? This question.
Is there any method for identifying fungi which is poisonous? Okay, I think we can look into the biomarkers. So whatever fungi we have already understood from the, classical system, or even from the DNA based, from the genetic based analysis also. So we can look into the biomarker if we are looking into the molecular identification. If you're going into
Exactly.
morphological, I think sometimes it will be very misleading because based on the citation, some fungus might be poisonous, and then it doesn't look like poisonous. And because citation can vary from human to human, right? I don't know if I answer it correctly. With the molecular marker only, we can, I think we can tell for sure that which is poisonous and which is not poisonous.
Yeah, it is just like identifying the aflatoxin producing fungi.
Right, sir.
Sometimes the gene will be there, just like as you told, DNA is not a good marker,
Right,
transcript or the protein. So looking for a toxin production,
right, sir.
you know,
Very well
by
said.
yes, TLC or HPLC method.
Right, sir.
Yeah. And if you are differentiating a large number of samples, then DNA fingerprinting looking for the toxin genes.
Right, sir. Very well said, sir.
Exactly.
Okay. So now we come to the end of today's session. So I sincere thanks to everyone who joined us today for this enriching session on from the 17th century to the 17th February 2026. New frontiers of microbial life. First and foremost, I extend my deepest gratitude to Dr. Swatantra Kumar
Thank you,
for
sir.
his insightful, inspiring, and deeply engaging lecture. And truly, your expertise and clarity has expanded our understanding of the microbial evolution. We are truly grateful for the time and knowledge you shared with us, sir.
Thank you very much, sir. Honor is mine.
Thank you so much, sir.
Thank you for giving this platform. Really,
I would
thanks.
also like to express my sincere appreciation to Dr. Sally Sharma for
Definitely.
her introduction to Dr. Swatantra Kumar and for her continuous support in approaching us. And only due to her initiative and involvement today, this played a key role in the making today's events means possible. So thank you, madam, for approaching us and to introducing the Dr. Swatantra Kumar to our society.
Most welcome, sir. And thank you
Thank you,
for
sir.
taking out time from all those discussions so that we could have sir's talk today. So I'm thankful to you for that, sir.
Thank you. And also my thanks extend to our respected All India President of MBSA Microbial Society, Dr. Arvind Deshmukh, sir, because of his guidance, encouragement, and support, which continuously inspire us to organize the impactful academic programs under the umbrella of Microbial Society. And also the special word of appreciation goes to all the members who joined today's session. My special thanks to our respected Dr. Halami sir, who is the main pillar of the roundtable discussion because he joined each of the session and he always support, or you can say encourage us to conduct the more scientific events under the umbrella of Microbial Society. So thank you everyone once again and for joining today's session. So we look forward for your participation in the future scientific activities of the Microbial Society. So thank you everyone for joining today's session.
Thank you, everyone.
Thank you so much, sir, for
Thank you.
today's session. Thank you so much.
Thank you, Dr. Kumar.
Thank you, sir. Thank you so
Thank
much,
you.
sir.
You have a
Great.
great time.